I would now like to welcome Dr. Donna Reece, excuse me, shh, merci. It works well. Try it at home. Shh. Okay. Dr. Donna Reece, Program Director for Multiple Myeloma and Related Disease at the Princess Margaret Hospital, and Chairholder of the Molly and David Bloom Chair in Myeloma Research, and co-founder, of the Myeloma Canada Research Network. Dr. Reece will talk about clinical trials and how important it is to bring new therapies to our patients. Dr. Reece?
Thank you very much. It's a great honor and a great privilege to be here and to see the excitement, momentum, optimism that has been characteristic of our field, really even over the last year or two. So today I'm going to discuss what's involved in clinical trials, because this is how we can most often bring the brand new drugs to our patients well before they are approved by Health Canada and are funded, which is another hurdle that varies according to province. So I'm going to cover in this brief discussion a bit of background about the drug regulatory process, very little bit about drug development, because of course if we don't have a new discovery, a new drug, then all of this is not pertinent. A small amount about the regulatory approval processes, such as Health Canada, which has to vet every new clinical trial application. And then at the local level at each hospital, there's a research ethics board or institutional review board that reviews the drug or the combination not only for its scientific merit, but also these boards consist of lay people, patients, lawyers, ethicists, perhaps people who are faith-based, to be sure what we are doing makes sense. Each trial has to be formally opened at the hospital, with training to the pharmacists, the nurses, the physicians involved, and that all has to be documented. And then finally, I'd like to summarize with some of the advantages for clinical trials for those people living with myeloma. So it was really almost 100 years ago that the Department of Health was established in Canada to actually regulate the quality of food and drugs. And by the early 1950s, manufacturers were required to file a new drug submission before they could market their drugs. And then the early 1960s changed things dramatically because there was the thalidomide fiasco. And now drug manufacturers are required to submit efficacy and, in particular, safety data to obtain what's called a notice of compliance, which allows the drug company to market its drugs. And then the regulation of drug products before and after approval requires really a complex set of legal and legislative requirements that has to satisfy both pharma that makes the drug and the consumers, that is, the patients that are using the drugs. There are underlying ethical principles which are driving clinical research, and I've shown what's been established by the multidisciplinary panel a number of years ago in the U.S., and Canada's is even a bit more specific, that underlying clinical trials, there must be a respect for human dignity, for free, informed consent without coercion, protection for vulnerable persons who may not understand what they're getting into, respect for privacy and confidentiality, justice and inclusiveness, and of course, minimizing harm and maximizing benefit. You just can't open a trial because you're curious if the drug hits a certain target in the cancer cell. Now, of course, as I mentioned, if you don't have a new drug to study, nothing will go forward, and there has been an incredible amount of research underlying drug development in myeloma specifically, and I just outlined here one way this can happen to give you a flavor. in myeloma or other cancers, one needs to find a vulnerability in the cancer cells which have an advantage in terms of life expectancy, ability to survive, and to proliferate. So if you can identify a vulnerability, then you can design a drug to go in and hit that Achilles heel. And this new drug or this new compound, this new molecule must be tested in myeloma cells and then animal models. And you validate that this target actually makes a difference in a person. And then you can move towards offering the drug in a clinical trial. Now, I'm not a scientist, but there is a way to do this. Particularly, I'll show you an example that's being done in several centers and in our own, where you look at the genes, or actually their RNA product that codes for a protein that can enhance myeloma cell survival. And you're looking for, using a robotic platform, genes that if you can inhibit their product, the cells may die, the myeloma cancer cells. So this slide shows an example of what happens, that each of these yellow long lines represents a cell line or patient samples, and all the yellow genes that if you knock them out or inactivate them, which means their protein doesn't make it to do its job in the cell, nothing happens, but you can see there's a target identified of some highly effective drug targets that can then be taken down the process I've just described. So this requires some smart people with some really sophisticated equipment, and then the ability to take that target and create a safe molecule that's a drug to help our patients. So before we start a trial, I've alluded to one way of drug discovery. You have to test it in cell lines, you have to test it in animals, and then you have to work out what happens when you give this drug to a person. How long does it last in the body? How is it eliminated from the body? Through the liver, breaking it down, through the kidneys, eliminating it, and then, of course, the toxicity. What's the early toxicity? And then what if you give the drug for many months? There are surprises. Is there late toxicities? Is this a drug that can be given as a pill? Does it have to be injected intravenously, or can it be given under the skin subcutaneously? And of course, what's the right dose? So as we move along the drug development pathway, I've shown here the preclinical research. Then there's clinical research, which I'll describe in a minute, what we call phase 1 trials, phase 2 trials, phase 3 trials. The data at some point, if everything's successful, is submitted to Health Canada, and then there may be post-marketing surveillance. And then the important caveat that in Canada, because of our healthcare system and the funding of these drugs, Health Canada approval does not necessarily mean the drug is funded. There's another layer you probably heard about earlier this morning, that it has to be vetted for value by each of the provinces before it is paid for and offered hopefully universally. So I'm going to spend the next few slides going over phase 1, 2, 3 trials, because I think if you or your loved one is deciding to participate in a trial, this information is really important to know about and to ask of your physician, research nurse, and the other people at your center that may be approaching you about participation in a trial. So a phase 1 trial is mainly based on safety and to get the kinks, if you will, out of the dosing, and we look for clues. Is it working? And I'll give you an example of bortezomib approval in a minute. A phase 2 trial, of course you're interested in safety, but you really want to know if it works in a particular disease. And then a phase 3 trial is your randomized trial, and of course safety's monitoring, but you're comparing it with a standard. And then there are some phase 4 trials, but that's really not too relevant today. So phase 1 trial is often a dose escalation study where you start out at a dose that's very small for safety, and that dose may not be effective. And there is a process to slowly go up in cohorts of patients to determine the maximum tolerated dose, or what we call in the jargon MTD. We're also, of course, looking to see how well it works. But if you don't know the right dose and the right interval, you really can't, with intelligence and safety, go on to a phase 2 trial, which uses the maximal tolerated dose for one disease usually. And the parameters that are measured, of course, the response rate, how much does a tumor shrink, time to tumor growth, again, or what we call the time to progression, the progression-free survival of the study drug is of particular importance for Health Canada, for funders, and of course patients. And it means how long patients survive without their disease growing. Overall survival is the endpoint of all our studies, but fortunately in myeloma, people live many, many, many years. So there's this discrepancy. Health Canada wants survival data, but there's a recent proposal of a trial for maintenance after transplant that they're expecting the PFS to be an average of 8 years, and the trial will take 10 years to complete. So you can see how that may not be relevant to us because the field is going to be dramatically different in 10 years. So this is the science of a trial, but this is not really what we need to help our patients tomorrow and make decisions. And of course, toxicity. Phase 3 trial, as I mentioned, is a randomized trial to show that the new drug or combination is equivalent to or better than the previous standard, and this is really what Health Canada seeks. Occasionally, the phase 2 trial information is stellar enough in a situation where there is an unmet need, there is no other good comparator, there will be approval. This is more often in the U.S. that a phase 2 trial will drive approval, but it has happened in myeloma in Canada. So as I've mentioned, the important endpoints are PFS and overall survival. Just to go into this process a bit more, a phase 2 trial is usually fairly small. It's 1 to 3 sites, and there's often a 3-by-3 model where the statistics are written that 3 patients are entered into a trial, then you stop entry. And you monitor those people for a certain amount of time very carefully to see there's no side effects. If one patient has worrisome side effects, and there's stop criteria built in these trials, one patient has side effects you're concerned about, you can enter 3 more patients. So you have a cohort of 6 patients. If only one has worrisome side effects, you are allowed to go to the next dose. So these trials start and stop, and while they're very good for getting brand new drugs for patients, they're slow. And they aren't randomized dose escalation.
We weren't here this morning, that's okay.
Okay. All right. All right. I hope they're fighting the myeloma cells. So a phase 2 trial is somewhat larger, usually around 60 to 70 patients. And here they're limited, let's say, to myeloma. These are usually multiple sites. Usually we know what patients are given. It's not randomized, and it's fairly short in duration. And this is particularly, I think, important for our patients, because most of the time everyone gets the new drug. There is not a comparator to an old standard. Then, as I keep talking about the phase 3 trial, these turn out to be very large. The ones that have approved the new drugs like bortezomib and lenalidomide and pomalidomide were hundreds of patients. They usually involve several countries. And they may be randomized. For instance, with many of the trials in the past, the control arm got high-dose dexamethasone, and the test arm got dexamethasone and the study drug. So these are a little bit harder sell, I think, for those people who want the new drug. Sometimes there's a way to get around that. Sometimes, and as investigators, we like that for our patients, and I think our patients find it more attractive, is if the control arm isn't doing well, their patients are allowed to cross over and then get the study drug. Now that makes it a little bit harder to tell survival, which is what the regulators want, but I think it really makes things more attractive for our patients. So we push for that if we can. I'm going to give you an example of bortezomib development. This was over a decade ago, but when bortezomib was first developed, and you probably know it was developed by a Canadian, and it was tested in these centers here over a wide range of doses in a set of different dosing schedules, how many doses per week, per month. And then what they saw here, these were done in lung cancer and other solid tumors. When they got to the heme malignancies, it wasn't very many, but they saw activity at these doses, particularly in myeloma, Waldenstrom's macroglobulinemia, and lymphoma. So then this drug was taken forward. I won't go into the phase 2 trial, but what was called the APEX trial, which was really the first large randomized trial in relapsed myeloma. And it had hundreds of patients that either got bortezomib as a single agent or high-dose dexamethasone as a single agent, and the rest is history, because of course this was the winning arm. So if we want, let's say, MCRN or a company wants to develop a clinical trial with a new drug or a new regimen, you have to design the trial first and write the protocol, which is like a 100-page recipe book with guidelines, background of the drug, how you dose it, precise instructions for what you do to reduce the dose if there's problems, what the endpoint is. And it has to be feasible. You can't do a drug on myeloma if you don't have anyone with myeloma living in your country. And funding. Who is paying for all this? Clinical trials are expensive. I'm going to show you the background of who's involved in these studies, but I would say really pharma has to be the funder. And if the study is coming from pharma to get drug approval, that's a different situation than if an investigator or an organization like MCRN goes to pharma and say, will you help us do this trial, which is not going to get the drug approved, but we think it's a great idea and we think it will help patients. And our pharma partners do have provisions to fund these, what we call IITs, or investigator-initiated trials. The trial for, let's say, a phase 2 trial, just to give you a broad guideline, depending on the complexity, usually costs between $2 and $4 million. So this is a lot of money to try to raise. And for investigator-initiated trials, because of the nature of this, the amount of money usually doesn't cover all the costs. And these are trials that we may need to dip into donated funds, backup funds, to actually get them to our patients. Once the trial is written, it's submitted to Health Canada for approval. They have a 30-day turnaround. If they have no objections, then we can proceed. If they have questions or concerns, those need to be addressed. The trial is then submitted to each center's research ethic boards. They need to prove the consent form, that it's understandable, it describes everything, how many extra visits, every single toxicity that could happen. They are usually daunting for a patient to read. We try to highlight what's realistically been seen with this drug and point out that if you look at the side effects of aspirin in the usual information sheets, there's a list of 100 complications too. Then each trial has to be formally activated at each site with a visit. We do measure metrics. How long does this process take? Patients are waiting. And I'll allude to that a bit in a minute. And once the trial is going, there's continuous monitoring. There's safety alerts for every side effect that's significant of each drug in a combination that goes to the center and has to be reviewed and, if necessary, kicked up to the ethics board at the institution. Of course, Health Canada has an eye on all of this. And there may be audits that come unannounced to be sure all the regulatory and safety work has been done. So I've I think, covered most of the trial components here, and I've left out though one important one, there needs to be usually some connection between the sponsor and the sites, and there is often a company hired called a CRO, a clinical research organization, that makes all this happen. I think the ultimate goal is MCRN. We can be our own research organization that does this, because this is obviously usually a private company, and there's a lot of money involved to pay the CRO. I've also left out that contracts are needed between the institutions and the sponsor or the CRO every step of the way. So the legalities of this to protect everyone involved are not inconsequential in terms of complexity or time. And when we look at our metrics, it's the contracting that actually holds things up. Here's a summary slide of the process again. Health Canada approves the trial that's been submitted by a sponsor, cooperative group. Each board, let's say in Montreal or Vancouver, then activates it. The CRO brings it all together, and then patients can be offered this. How long does this process take? I can tell you our current goal metrics, median, is from when the trial is submitted is 8 months. So that's considered a good amount of time. So I'm going to talk a little bit about what happens at the trial site. This is where you will have your interaction with or interface with the clinical trial. There's a local physician who's the local principal investigator who's responsible legally, ethically, and scientifically for the conduct of the trial, and then the essential clinical trial coordinator or research nurse, and the patient is here. At Princess Margaret, for example, we have about 36 active trials per year for myeloma. We enter, this was 2004, about 75 patients onto trials, but then patients, fortunately, if the trial works, may be on these trials for years, where they're seen monthly. All the paperwork and data for each visit, any side effect is continually collected, submitted, and reviewed. So we run between 165 and 175 patients per year on study, and it takes 17 people to do this. A research manager, clinical coordinators. The level of paperwork is incredible. Research clerk, we have to track every invoice that comes through, and then our 5 research nurses. I have to emphasize that none of these positions are covered by the healthcare system. None of them are covered by the hospital. So we have to pay for these salaries, and you want good people to do this, either from the trial finances, which unfortunately often don't cover this, to donated funds. So talking about just a couple of more topics, one of the things that I think is very relevant for patients is when it comes down to the site, there are these inclusion-exclusion criteria. They're mainly required for safety, but they're also required so that the trial answers the questions that it's supposed to, because if the data doesn't tell us that a drug works in newly diagnosed patients versus advanced disease, the data is meaningless and you can't take it forward. But this can be a real stumbling block for patients because it specifies how many lines of therapy, the blood count requirements, neutrophils or the infection-fighting white count may go down with different lines of therapy, platelets may go down. If the drug is excreted by the kidney or liver, these tests have to be normal. There have to be fertility concerns so that an individual doesn't become pregnant or make someone pregnant while receiving certain drugs. So we are always fighting that we want people with low blood counts to be allowed, because we can certainly transfuse if necessary or give growth factors like Neupogen or EPO. What are the advantages of a clinical trial? There's the benefits to society to get the drug on the market or help others, but I advise my patients, do this for yourself. You know, you don't owe anybody anything. So I think the main advantage is getting access to drugs that are not otherwise available. Either they're not on the market yet in Canada, because the pharma companies get the drug on the market first in the US, second in Europe, because there's a body that approves most of the countries, and then Canada, Japan, Australia are in the third tier. And then they may not be funded, even if they're approved by Health Canada. And I think another real benefit, at least at Princess Margaret, where we have about 8,000 myeloma clinic visits a year. It's very easy to get lost in this center. Having a research nurse is, I think, a really, really nice advantage, because she or he is dedicated to being receptive to your side effects, expedite your visits, make sure everything happens. So I think you're going to hear about Myeloma Canada. This is our organization that has been brought in under the umbrella of Myeloma Canada. The goal is to get new drugs. These are mainly going to be phase 2 trials from coast to coast, because not everyone lives in Montreal or Toronto. And we have a nice network across the country, and we have a very exciting menu, if you will, a portfolio of trials, some of which have already been started and some are in various stages of planning, negotiation with pharma to get the funding to do these drugs, and then the activation. At least at Princess Margaret, of these 30 plus trials we have per year, some of them come from the cancer, what used to be NCIC. We do have associations with the MMRC in the US. Industry funds most of the trials, but we're bringing in the MCRN trials. This slide shows the different categories of drugs that I don't have time to go into. You'll recognize your alkylating agents, steroids, your proteasome inhibitors, your immunomodulatory derivatives, the new monoclonal antibodies, brand new drugs, and the immune treatments. And I've shown with the arrows what we have for trials that people can go on to now at Princess Margaret. I haven't included the ones that aren't allowing patients to start treatment, but they may be on treatment for elotuzumab or some of the others. But this is what we have on our agenda for MCRN trials with the new agents, which I think is incredibly exciting. So I hope this has given you a bit of understanding of all the background for clinical trials, and I would say that Myeloma Canada, under Aldo and the other leadership, has pushed for this to be part of Myeloma Canada's spectrum of pillars, in addition to education, awareness, advocacy, the support networks. But I think the next step has been get these new drugs to our patients safely as soon as possible. So thank you very much.